So let us start. Dr. Vidya madam, are you online? Thank you, madam. Now, present it will be the back and the management. Scientist from ICFRI. She mainly works on the ecosystem, factor resistance, I mean the micropathology interaction, and how to control the using probiotics, etc. Dr. Vidya.
Yes, sir. Respected Director, CIFRI, my senior colleagues and friends, I am Dr. Vidya, scientist at Aquatic Animal Health and Environment Division, ICFRI, located in Chennai. Today, I will explain to you about the importance of innovation is the specification of a lower invertebrate. It has been found in more than 50 countries around the world. It's very risky, but it gives us high income. Profit is more. There are a lot of risk factors associated with this particular farming. The major inputs for this shrimp farming is the water, the seed, and the feed. As you know, the feed quality, which is already standardized and commercialized, so the risk factors associated to this feed will be very less. But compared to the water and seed, where the major risk factors are lying over the water and the seed. So the most important risk factor during the farming is the disease occurrence. It can be due to bacteria, it can be due to virus, microsporidium, or many other things. And if we look at the risk factors other than the disease agents, the poor water quality, which aggravates the disease by stressing or altering the physiology of the animal which is farmed in the water environment. So this is a typical shrimp pond ecosystem where you can see a water phase, and it was covered with a shrimp bottom, pond bottom, and the shrimps are inhabited in such an environment. Over the farming, example, the shrimp farming is done for 3 to 6 months. Over this run, we will get a lot of feeds and molting shells. It will sink to the bottom, and the bottom will be always filled with this type of fecal matters, organic matter, and which will be in turn a source for the bacterial multiplication and germination, and many other parasitic related problems. And what all the things we regularly give to the shrimp ponds is the feed, as well as some other probiotics we also use for the management of this system. So this is the general aspect of a shrimp pond ecosystem what we are going to deal with. First, I will tell you about the water quality in shrimp farms. There are about 17 quality parameters which are fixed for these shrimp farms, and there is a optimum range for these all quality parameters, starting from the temperature, pH, salinity, total hardness, total alkalinity, transparency, total suspended solids, dissolved oxygen, COD, BOD, total ammonia, free ammonia, nitrite, hydrogen sulfide, nitrate, phosphate, and primary product. All these factors have an optimum range for the shrimp farms to be successful. I'll explain you each one. Dissolved oxygen, the other gaseous materials in the shrimp pond are potential stressors. For example, dissolved oxygen, carbon dioxide, ammonia, and nitrate. All these are potential stressors to the shrimps, and very important during the growth period. And the other parameters like salinity, pH, alkalinity, minerals, turbidity, and nutrients, which affect the plant productivity within the primary productivity in the pond where we are culturing the shrimps. And it in turn affects the aquaculture production. So SIBA has developed a multi-parameter testing kit which is helpful for the farmers to find out the pH, carbonate, bicarbonate, total alkalinity, dissolved oxygen, ammonia, nitrate, calcium, magnesium, and total hardness of the water, where we are measuring our shrimps. So first, let me begin with the source, where we are taking, from which part we are taking the water for shrimp farming. Usually we take water from the creeks or from the through a pump from the sea in the tidal zone of the seashore. So these water are taken into the shrimp farms. Now, when we select an area for a shrimp farm, we should look at the creek are not receiving any agricultural runoff, industrial waste, or any other such type of effluents. Otherwise, it may contain lots of pesticides and other pollutants which can come into the shrimp farms. So when you select this farm or an area for shrimp farming, we should look at the creeks or the area where least occurrence of these discharges possible. So this is a typical representation of a shrimp farm, where the lower part indicates the estuary or the creek from where we take the water into a unit. It is called sedimentation unit, because the water which let into the shrimp farms, the sedimentation unit will be containing a lot of suspended particles, and all those things will come along with the water, will be there in the, if you directly take into the farm, it will contain contaminated water system. So for Banami or any other shrimp farming, we need to have a sedimentation unit first of all, followed by a reservoir. The sedimentation unit will allow you, the sedimentation unit will allow you to settle the whatever suspended particle is there in the water, which will allow this unit will allow it to settle. Then from there, the more clearer water will be taken to a reservoir that will feed the production units in the shrimp farms. So in the reservoir, the typical that area is demarcated for the reservoir pond, where the pond will the water will be given to treatment, and it will be the treated water only will be taken to the production units. And at the corner or at the other side, there will be an effluent treatment pond or unit, where whatever the water which has comes out of the ground will be treated and released back into the creeks. That ensures the safety of the environment, so that the other people can also utilize this water when it comes to the creeks. So the water which takes in and comes out of the shrimp farm should be of good quality, or we have to ensure the quality and safety of the environment. So what are the treatment we give when we take in the water to a reservoir pond? The direct use of creek or seawater is risky because it introduces many pathogens. Can be bacteria, virus, or microsporidia, or any type of pathogen. So we have to eliminate that type of risk. So what first of all we have to do is we have to pass through this water through a 60 micron mesh. There are different types of micron meshes that are available, but here we recommend 60 micron mesh to the reservoir pond, so that the carriers, many of these small animals living in the water can be carrier for many viruses or bacteria. So in that case, if we use this micron mesh, we can eliminate such type of carriers to this system. And before starting the shrimp farming or the farm unit, you have to remove the silt in the pond bottom. There is called a set of protocols for the pond preparation anode. So that I will be telling you later. Now, so the suspended solid should be removed by settling. The water, whatever suspended solid, after this screening, 16 micrometer screening, the water will be contained in suspended solid. So that should be the water should be allowed to settle, so that the suspended solid can be settled down to the sedimentation unit. Then this water will be taken to the reservoir, and we can give it chlorination. That is the treatment part, so that we can disinfect our water before entering into the shrimp grow-out ponds. So this treatment we used to do with sodium or calcium hypochlorite. It's called chlorination, and we can use less than 100 ppm. And here I mentioned at the rate of 30 ppm. That means 150 kilogram per hectares. So the calcium or sodium hypochlorite which you use may have the activity percentage will be varying. So depending on that, we have to calculate the exact amount required for the I mean, like for a hectare, for an acre, like that we have to calculate. For example, if you are going for a 10 ppm chlorine and you have 1 hectare pond, you have to use 100 kg, and your activity of the chlorine in that is 30%, means you have to multiply the 100 kg into 100 by 30. So you have to use the 333 kg of the sodium or calcium hypochlorite for your water, which you have taken in. And also, I have told that the creek's water can be containing the heavy metals or pesticides, because it may be contaminated with the agricultural effluents or the sewage. All those things can come into the creeks because all these things are interconnected water bodies. So it is best if you check or analyze your water for the heavy metals and pesticides, because heavy metals and pesticides really toxic pollutants which come into the animal, which in turn come to the human. So it is very dangerous. So first of all, before we have to check the source water for these heavy metals and pesticides. So once the chlorination is over, as per the calculation, we have to allow the chlorine to act for 12 to 14 days. So a fortnight we can keep, that means 2 weeks we can keep. Then at the end of the 2 weeks, what you have to do is, we have to remove the residual chlorine from the water. So we have to aerate this water for 48 hours. And even the chlorine is not removed, we have to add 1 ppm of sodium thiosulfate or 1 ppm of chlorine. That will neutralize the residual chlorine levels in the treated water and expose this water to the sunlight. After that, we can use. And this water should contain less than 0.001 ppm of chlorine. And another factor is the turbidity. This water may contain a lot of clay particles. We cannot avoid that. So the sedimentation can be done. If it is not possible, we can use alum at a rate of 5 to 7 ppm. So water is important to us, the soil parameters. So before the construction or when we construct the shrimp farms, the soil must be tested for the following parameters. There are so many labs are available for testing soil and water quality parameters. Even SIBA, we are developed kits for soil and water. So all these parameters have to be checked prior to the construction of the farms. For example, pH should be of 6.5 to 7.5. When we look at the soil parameters, there is a lot of influence it can make on our shrimp farming. If it is an acid sulfate soil, if the pH is less than 6.5, it can affect the pH. Further, if you add the water, it can reduce the pH levels in the water. So if continuously, there will be lot of maintenance cost is incurred during the farming. So it is better we should check the soil parameters prior to the construction. So such soils can be avoided. So best soil structure can be used the sandy clay loam and clay loam. And all these parameters prior to the starting of farm, we have to check. Should be an authorized lab. Next one is the dissolved oxygen. It is the most critical and limiting factor in the aquaculture. The shrimp farm can get, the pond water can get the oxygen through diffusion or it as a byproduct of photosynthesis, and it can be lost from the water body through the respiration of the animals or aerobic decay of the organic matter. The optimum level of oxygen is more than 5 ppm. And the low dissolved oxygen has a direct influence on the animals. It reduces the feed intake, it leads to the slow growth, it makes the animals susceptible to diseases, and it totally changes the metabolism of the shrimps. It has the indirect influence on the water quality parameters such as, the oxygen, the gases present in the water, that in the pond water, that is unionized ammonia, hydrogen sulfate, methane. All these have an impact, interrelated with the dissolved oxygen levels in the water bodies. So the safe levels of the dissolved oxygen in a shrimp pond is 5 ppm, more than 5 ppm. That is the best condition for the shrimps. 1 to 5 ppm is stressed, will causes slow growth, it increases toxic substances, and as I said, it will not grow, it will not eat, and susceptibility to the diseases. And less than 1 milligram per liter, even in for more than a few hours, is also lethal for these animals. So in a pond system, if you monitor the dissolved oxygen levels from morning to the whole 24 hours period, you can see from morning to the midnight after 12 p.m., the oxygen can be maintained well above 5 ppm. But when it comes to the midnight to the morning time, like 12 a.m., 1 a.m. to 6 a.m. So this area is very critical. This time period is very critical for a shrimp pond, because at this time only the dissolved oxygen comes down. The main reason is during morning to that 12 a.m., that 6 a.m. to the 12 a.m., the phytoplankton will generate oxygen. After that, it gets light and it uses for photosynthesis, and oxygen is released. But when it comes to the night time after 12, the photosynthesis stop. So they start using up the oxygen for their own purpose, the respiration purpose. So that period, the oxygen will be reduced. So it's very critical to use aerators during the night hours to get maximum oxygen to these farms. The dissolved oxygen can be altered by temperature and the stocking density in the shrimp farms, the biomass in the farms, and as I said, highest during the day, and the dissolved oxygen will be lower at night, so provide the aerators during that time. So how aeration helps for dissolved oxygen? Aeration helps by mixing the water at surface and the bottom. Imagine a stagnant body where we are providing aerators. So this will mix up the surface and the bottom waters so that the dissolved oxygen can be sent to every part of the pond. And the aerators is also so placed that there should be continuous movement in the pond, and water should be mixed up pond so that the dissolved oxygen maintains all throughout the system. And one more thing is we have to remove the black mud from the interface of pond water and the bottom soil, because this part is having a lot of organic matter, leftover feed, and the molting shells. Everything will become and accumulate because of the action of aerators. So this part should be placed with a central drainage system underground, so that periodically we can remove this sludge, which is harmful to the farming, can be removed from the pond bottom easily. So this central drainage system is in most of the cases now, people use the central drainage system to remove the sludge during the culture period. So come to the aerators. 1 horsepower aerators is required for the 300 kg of biomass. That is the range fixed for these aerators. And as I said, the aerators should be adjusted so that all the area should get the equal movement and mixing. And how we can measure the dissolved oxygen? We can use automatic dissolved oxygen meter, and there is a titration method also. We have made a kit based on that. It's based on Winkler method. So this is available for use for the farmers. So next is salinity. We know that shrimp faces urihalane. Basically, it's 30 ppt. Salinity, where the seed produced in 30 ppt salinity exactly. So these host larvae, which is reared in 30 ppt salinity, are adapted to the lower salinities and brought to the shrimp farms. And the salinity means the salt concentration of the water. So when we take up the crayfish or the sea, the salinity of these areas varying with the source of water as well as season. During rainy season, there will be much drastic change in the salinities. And each species what we use for the farming. For the Pinnace monodon, Pinnace vaname, the salinity ranges vary. And this salinity is a very important role in growth, reproduction, migratory behaviors, and metabolisms. And the apt The optimum salinity range for Pinus monodon is 15 to 30 ppt. For Vanami, it is 2 to 40 ppt. You can adapt Vanami to such a range that can be utilized in varying salinities. There are mitigations available for inland saline waters and low saline waters. Many people are applying minerals and doing the farming of Pinus Vanami. So it is possible in low salinities also with mineral addition. So the salt can be checked with a salinometer or refractometer, where you can drop a small drop of water from your farm, then view through that. The blue color will indicate the margin of the blue color in the lower part will indicate the ppt salinity of your water, and then wash with fresh water and again reuse for some other tones. So pH is the measure of hydrogen ion concentration in the water, and it tells how acidic or basic the water is. Usually, the alkaline side is preferable for the animals because this alkaline water will resist. It has the buffering capacity. The pH can be altered by acidic source of water and acidic soils due to rainfall, poorly buffered water, that is low alkalinity waters, stocking density, feeding and rate of sludge formation, presence of micro and macroorganisms, phytoplankton and rate of carbon dioxide production, and the respiration of the aquatic species. And if pH can affect the pond fertility and quality, plankton growth, and metabolic concentration. The optimum level of pH is 7 to 9, and in a diurnal period, like I told now, in the pond there is a plankton productivity, so it can vary with the plankton production. So in a diurnal cycle, there should not vary more than 0.5 units in the pH. So the suboptimal pH can make stress, increase susceptibility to disease, poor growth, and low production. And the causes of suboptimal pH: acidic water, soil, acidic sulfate soils, poorly buffered water, high alkalinity and low hardness, acid rain, etc. are the causes of suboptimal pH in water bodies. How can we reduce? Lime application and dolomite at the rate of 80 kg per hectare at night, weekly twice after 20 days of culture. We can use this dolomite for mitigating the pH. Then to reduce the pH, we can do the water exchange, apply jaggery, fermented juice. That fermented juice is made of rice bran and molasses. That we can add 30 kg per hectare to reduce the pH if it is in alkaline side. The pH can be checked with a pH meter, and a titration method is also available, and we can check the pH with these two methods. Temperature is essential for metabolic and physiological activities and life processes such as feeding, reproduction, movement, and distribution of aquatic organisms. And the optimum level of pond temperature should be 25 to 30 degrees Celsius. And operation of aerators during warm and calm afternoons helps to break thermal stratification by mixing warm water surface with a cool subsurface water. This thermal stratification is a common occurrence in the water body. So this will not allow the proper mixing of the water. So that time we can operate the aerators to break the thermal stratification. So the temperature can be measured. We know the normal means of measuring temperature. The next one is turbidity. Turbidity can be measured in a pond using a SH disk. Turbidity by plankton is desirable because it enhances the primary productivity and increases the aquaculture production. Turbidity measures the optical property of water. It causes light to be scattered or absorbed rather than transmitted through the water. So the turbidity due to clay will be harmful. So it is undesirable. It can affect light penetration, plant growth, destroys benthic organisms of flavored shrimps and algal blooms. So if turbidity is high, it can even kill the animal. So the disk measurement, if it is above 60 centimeter, means it indicates the poor plankton density which needs to be fertilized. If less than 20, that means the high plankton density. So we have to reduce the rate and frequency of fertilization. The optimum value should be between 25 to 50 centimeter when we measure. How we can mitigate the clay turbidity? It can be done through the flocculation or percolation. For this purpose, we use to add gypsum, that is calcium sulfate, which tetrasulfate, which is at the rate of 400 to 1200 kg per acre we can add, and other one is the alum, which can apply at the rate of 60 to 110 kg per acre. The alum is the most effective in controlling this clay turbidity. And the total alkalinity, this is the ability of the water to neutralize acid without changing the pH. This is the buffering capacity of the water, which denotes the amount of carbonate, bicarbonate, and hydroxide ions present in the water, and determined by the type of water and the nature of the pond bottom. Sandy soil and organic soil will provide you low alkalinity, so it should be taken care of. The productivity can be improved if we increase the total alkalinity. The productivity can be improved because it supplies the carbon dioxide for the primary productions. If pH is high and the total alkalinity is high, there will be a rapid decomposition of the organic matter. So the best the concentration for total alkalinity is 75 to 300 ppm. That will be ideal. More than 300 is stress, and less than 75 is a stress to the farming animals. So if alkalinity is low, we can apply lime or dolomite at the rate of 180 to 300 kg per hectare at one time at night. If very high alkalinity is prevalent, then if it is less than 20 ppm, pH beyond 11, it can cause mortalities in the farm animals. And EDTA at the rate of 20 to 30 kg per hectare at night will be applied to control the total alkalinity in the safe range. So this is the one which is used in the SIBA for determining the alkalinity. It is available with the kit. Individually also we can make it, purchase it. And total hardness, it indicates the amount of minerals present in the water. So these minerals are important for the growth and metabolism of the animals. And among the minerals, the ratio of potassium, sodium to potassium and calcium to magnesium in water are very highly important for survival, growth, and production rather than salinity. So this is the calculation which is required for different salinities of water. For example, for 1 ppt and 5 ppt and 10 ppt saline water, the requirement of minerals are given here. On the left hand, on the right hand side of the table, you can see the calcium, magnesium, sodium, and potassium in ppm levels, that is milligram per liter. So for 1 ppt, for example, for the calcium, at the rate of 1 ppt salinity in the water, you have to add 11.6 milligram per liter of calcium should be added to 1 ppt. For 5 ppt, it is 58 milligram per liter. For 10 ppt, it is 106 milligram per liter. So like that, for the magnesium, sodium, and potassium should be followed up. This is the range for the different salinities. So these minerals are not in actual form. It will be available in the form of fertilizers. So this, if the pond water is deficient in any such material, what we have to do is we have to choose any one of the. If, for example, I have given an example over here. In the case of potassium, so for in the previous one, we have seen that potassium requirement, and from that we have to calculate which PPT you are in. So take that, and you have to put at 200 ppm. So what you have to select is from these fertilizers. You have to select which potassium is in the potassium chloride, that is chlorate of potash containing 50% of the potassium. So you have to add 200, what you have to do is the 200 divided by the 50%. That 50% can be written as 50 by 100. So 200 divided by this much will give you the exact amount of potassium fertilizer to be added to the system. This way it has to be calculated. So this is the kit which CIFRI has developed to check the total hardness. And coming to the metabolites, ammonia, nitrate, and hydrogen sulfide are the major metabolites in the pond ecosystem. And the factors affecting metabolites are pH, temperature, and salinity. So when ammonia in water increases due to the decomposition, the ammonia excretion by the aquatic organism will be diminishes. The level of ammonia in their blood will be, or hemoglobin will be increasing. That it will lead to the stress to the animal. So the total ammonia level less than 1 ppm is harmful to the animals. And unionized ammonia should be less than 0.01 ppm in the water. So levels above this will be toxic to the animals, which is found in the water bodies containing ammonia and nitrate. Nitrate is the intermediate product in the bacterial nitrification. It is highly toxic to the fish and also to the shrimps. Nitrate toxicity increases with pH, decreases with calcium and chloride concentration, more toxic to low saline waters, and optimum value is less than 0.2 ppm in the water. Ammonia nitrate can be mitigated through water exchange, aeration, use of probiotics, bioflow systems, removal of algal bloom by flushing, collect sludges at the center of the pond, and remove through central drainage systems. CIFRI has made a product called CIFRI Moss, which can be utilized to mitigate this type of issues. Mainly ammonia and nitrate reduction can be done through the intervention of CIFRI Moss. Is a product developed by CIFRI. So these are the kits available for checking ammonia and nitrate. Hydrogen sulfide is another metabolized unionized hydrogen sulfide are extremely toxic, and it affected by pH, temperature, and salinity. It is produced in the pond bottom soil under anaerobic conditions, and this heterotrophic bacteria which is present in the pond bottom utilizes hydrogen sulfide and oxidize the sulfur compounds and excrete sulfides, hydrogen sulfides. This is where the hydrogen sulfide is forming in the pond bottom. The optimum range is less than 0.003 ppm in the water, and lethal is more than 0.01 ppm. How can we mitigate hydrogen sulfide? One is aeration, then water circulation, exchange to minimize anaerobic zones. We can apply lime, potassium permanganate, hydrogen peroxide, calcium oxides. All these can have effect on reducing the hydrogen sulfides in the pond. For example, another example is the use of iron oxide, which can combine with hydrogen sulfide and releases insoluble iron sulfides that can be removed. And pond draining and drying can be done prior to the preparations that will oxidize sulfides available in the pond bottom. So this is all about the water quality parameters, which is essential for the ensuring the quality of the water, which is in the optimum ranges for the growth and survival of the shrimps in throughout ponds. Next is the bacterial diseases in shrimps. So since the scientific farming has been started in 1970 till now, there are a lot of viral and bacterial diseases are reported from shrimp ponds and even from the shrimp hatcheries. And the most common ones are virus, but a few bacterial cases are also available. The major ones are the fibrosis, necrotizing hepatopancreatitis, and EMS. The most famous and fatal is the early mortality syndrome or the acute hepatopancreatic necrosis disease. Normally, the bacteria are controlled. Bacterial management can be done very easily, but in EMS case, there is a strain with a toxin-carrying plasmid was causing enormous death in the shrimps. That is only an exceptional case. Otherwise, the bacteria in the system can be controlled through the management and husbandry practices. What are bacteria? Bacteria are very tiny, single-celled prokaryotic organisms. They can multiply their own, not like the virus, which required a host. But bacteria can survive in their environment by their own and can multiply to a large number, millions of numbers in a short period of time. There can be culturable bacteria and unculturable bacteria. Culturable means we can grow them in the plates or media, anything like that. But unculturable means we cannot. They will survive only in their environment. We cannot take out from their environment and grow them. And these bacteria can be photosynthetic and chemosynthetic. Photosynthetic we utilize just like the plants. Chemosynthetic means instead of the normal photosynthetic bacteria, they use the chemical compounds for their energy and synthesis of their food materials. So there are two types of bacteria are present: photosynthetic and chemosynthetic. And among these bacteria, also there are aerobic. Among aerobic, it is obligate and facultative, which is compulsory. Oxygen is needed for them. Facultative means they can live with or without. Anaerobic means totally in the absence of oxygen. So the bacteria can be identified. If it is culturable, we can identify through different methods of staining. One is gram staining, and as it was staining, gram staining, the gram positive will appear the takes up the crystal violet color. The other one which will take up the pink color appears gram negative, the violet color appears the gram positive. Importance of bacteria. As we know, these bacteria are involved in many processes in the along the evolution of different animals, along the evolution of any type of animals. They are part and parcel of our life. In any animal, if you take up these bacteria are present in them. So they are also part of the biogeochemical cycles. They are part of nitrification, denitrification. They are decomposers, and they colonize the gut of the animals, even humans. They are used. They are screened. They are isolated, identified, and used in different industrial purpose. For example, fermentation, and they can be used as probiotics and production of antibiotics. So what about bacteria in shrimp farming? The shrimp farm has two different phases. One is in the hatchery phase, other one is the grow out phase. The shrimp hatchery, where from the adult, the brooder shrimps, the young ones are produced. It undergoes a development for a month from nuclei to the post larvae. What we use in the grow out is the post larvae of from 10 to 14 ranges, that is 14-day-old post larvae. So during the shrimp hatchery phase and grow out farms, they are supposed to colonize with the bacteria where they are exposed to. Their medium may contain those type of bacteria where they are exposed and they take up the bacteria internally. When in the shrimp hatchery or in the grow farm, they are exposed to such type of bacteria. So these bacteria are taken up by the animals and they colonize in the body. So normally, if the conditions are okay, optimum ranges of water quality, everything you maintain, these bacteria is found to be stable with the animals. Once the stressors, for example, I said in the water quality parameters, any of such stressors are coming into play in these hatcheries or grow out farms, that in turn stresses these animals and provoke these bacteria to evade their systems, and they takes up the host capacity to resist them. That's why the diseases are developed. Bacterial diseases are developed. So comes to the major disease causing bacteria. It's not disease causing. Usually Vibrio is a opportunistic pathogen, which in opportunistic bacteria, which only invades when the animal is stressed. Otherwise, the vibrios are very safe they are very safe along with the host. Once the stressors are set in, the vibrios also develops in enormous numbers and cause the different types of symptoms in the animals. The n number of vibrios are available. The major portion of the sea or the brackish water system may contain the culturable vibrios. There are more than you can count up to 100 numbers if you take up the vibrio species in the brackish water ecosystem or in the sea. And the major pathogens supposed to be considered to be pathogens are coming under the Harvey clade. mostly Vibrio parahemolyticus. It is because of only due to the EMS or the HPND. Otherwise, parahemolyticus is a very normal inhabitant of the brackish water environment. Vibrio harvae, but in the case of Harvey and Camberley, they are found to be active pathogenic role in certain diseases that I will be dealing with dealing in the next slides. They always the vibriosis which is caused by these species are mainly due to the poor hygiene status of the animal or the environment where they are living. So this is the first one of luminescent vibriosis, or it is called luminescent bacterial diseases, where the tank where the host larvae are kept appears like luminescent, the bluish green appearance of the water, and the host larvae appears to be fluorescent. So what is happening is this bacteria can glow in this is a petriplate having the luminescent bacteria. Vibrio harvae or Campbelli, we have Vibrio harvae, also Vibrio Campbelli, which glow, which are luminescent, and these bacteria when the animals are stressed, it will invade the system and it will totally filled in the filled up its gut, capitoprangis everywhere, and finally the animal will glow like anything. All the stages from nuclei to zoea to mysis to post-larval stages are not susceptible, and it causes 100% mortality in shrimp hatcheries. As well, when these post-larvae are taken to the ground for stocking, it can come to the forms also. So it makes the animal weak, opaque, and luminescent. We can see the abnormal. See, this luminosity can be observed only in the night. We cannot see this glow in the daytime. So when you wanted to see them, you have to observe the animals and the tanks in the night time. Then we can understand that the luminance bacteria are present in the water system, tank or deep pond. These bacteria can make the animal lethargic. It totally colonize the internal bodies, and they are motile vibrios. They have flagella to move about. So these are the important facts about luminescent vibrios. We have developed a phage therapy. Phage is a virus eating the bacteria. So ICIFRI has developed a cocktail of phages, is called lumiphage, and which is containing 4 bacteriophages, which are effectively against the most of the what we have collected 376 different types of bacterial host. So we have tried, and against all 376 bacteria we have isolated, and 4 we have found screened out of that the phages, and we have made a phage cocktail to treat this disease. That cocktail is called phage cocktail is called lumiphage, and we have developed it as a product, and we have commercialized it, and we have filed a patent for that also. And these vibrios, now you have seen the whole body is internalized with the vibrio bacteria. The other one is it can make cuticular lesions, like black colored lesions can be seen all over the body. That due to the hemocyte this melanization comes from the hemocytes. So these cuticular lesions are example of such type of disease is the bacterial shell disease, where you can find light or dark brown focal lesions, and it causes the necrosis of the appendages and black spots all over the body. These black spots are mainly caused by the melanin pigmentation produced by the hemocyte. That is an inflammatory reaction on the shrimp body. And sometimes the appendages and the perioports and the gills appears like red, so it causes a reddish appearance to the animal. So that also an indication of the vibrios infection in the adult shrimps. And you can also observe, many times we can observe black gill in the adult shrimps when you go to a farm, visit a farm. So this also due to the vibrio bacterial infection. So when these animals are infected, do not take up the food. The post-larval guts will be empty, motility will be reduced, and it will be distracted. So these are the symptoms of bacterial shell diseases. And the most important part of these vibrios is the infection in hepatopancreas and the gut. Hmm? Uh, if the gut or the digestive organ is impaired, means the animal cannot take the feed. Their stomach will be empty, or otherwise, if it eat also, the intermittent feeding only happening, and there will be gas filling in their in the stomach, and their because of the inactivity, the hepatopancreas, the central, the area where the greenish color on the head part. You can see from the outside there is a green color organ is there in the head part, central head part. That is the hepatopancreas. That will be darker, larger, and sometimes it is shrunken. And all these, all these, whatever the bacteria which affects the hepatopancreas and gut will release the digestive material in white color, or the gut will appear white color. So whatever infections happen to these two organs may have a symptom of white color duck or white color fecal matter. The most important diseases which is affecting the hepatopancreas and gut is the acute hepatopancreatic disease, which is caused by a vibrio, that is called Vibrio parahemolyticus, which has a plasmid which contains toxin like PIR-A, PIR-B. And these are some type of toxins come to this parahemolyticus strain naturally or accidentally. But this parahemolyticus having the toxin is causing the early mortality syndrome in different countries. It is causing 100% mortality in the shins, and stages postlarity from 20 to 30 DOC were susceptible and causing enormous mortalities in the shin farms. The clinical signs of these diseases were lethargy, slow growth, and the erratic stimming behavior, loose and short cells. And the major part is related to the hepatopancreas. It will be discolored, white, pale, yellowish discoloration of the hepatopancreas. It will be swollen and small, shrunken sometimes. And the gastrointestinal tract, the stomach, the tract will be empty, and the stomach will appear milky. So the symptoms of hepatopancreas and the gut will explain you where the shim is infected. These are the cross signs. The left hand side of the slide indicate the normal shim with a full stomach, with the pigmented, the greenish pigmentation of the hepatopancreas, whereas the EMS affected shim, you can see the white color central portion where you can see the intestine usually, but you cannot see it is like appeared white. Atrophied hepatopancreas, there is no coloration. When you compare, this is when we compare the color. So this is regarding the EMS. And next one is the white gut syndrome. This white gut syndrome, as we know that in HPND there is white gut. The similar conditions are present in our Indian conditions also, but it is not correlated with Vibrio parahemolyticus, but it is correlated with some other pathogens like microsporidiosis, which is caused by EHP. So in EHP and as well as the high Vibrio loads also causing white gut syndrome in Indian conditions, but not by AHPND. So this white gut syndrome is more visible, more than 300 grams, and it is noticeable with size variation also noticed in the shins. Mortality also reported, and this in the white gut syndrome is also called white feces syndrome. The farms which is affected may contain white colored fecal threads floating all over the ponds. That is an indication of white gut syndrome in the animals. So in white gut syndrome, we have seen that there are pathogenic vibrios, but it is not the AHPND strain containing a 0.2 to 10 raised to 5 colony-forming units of vibrio in their hemolymph. And it has been seen that it has a strong correlation with the endocytosis of hepatopenia. Of course, these hepatopancreas and gut are invaded by the EHP, and any infection on this part may cause a white color discut and discharges from the animals. Another one is most common in Indian shrimp farms are running mortality syndrome, otherwise called RMS. It causes the daily mortality of 2 to 3%. We can observe it from 40 to 30 DOCs. But in our study, INZIBA has recorded there is no involvement of any pathogenic organisms. But the histopathological studies have seen that necrosis and hemocytic infiltration in the shrimp tissues. This may be due to the increasing stocking density beyond the recommended level, and the cumulative effect of the pond environment during the culture might be causing the running mortality syndrome. It can also relate to the mineral deficiencies and the levels of different metabolites. So, how we can diagnose the bacterial diseases in shrimp farms? I am just stating with the help of the most severe disease, AHP, acute hepatopancreatic necrosis disease, otherwise called EMS. So we have the scientific scientist has developed 3 levels of confirmation. First is the isolation of bacteria by culture method, the classical microbiology. We used to isolate, how to isolate the bacteria by classical culture method, and identification by biochemical and the molecular methods. Next one is the parahemolytic strain, which causes AHPND. To confirm its presence in the animal, we have to do a histopathological technique. Since it is affecting the hepatopancreas, we have to excise the hepatopancreas and do a histopathology. The above one, which I showed here, is a normal histopathological structure where you can say perfect movement. Whereas if you look at the lower part where the hepatopancreas appears like gone, it's structure totally gone, necrotic and infiltrated with hemocytes. The total structure is lost. It appears like some type of toxic effect on this hepatopancreas. So through histopathology also AHP can be investigated. The other one is molecular detection for AHPND. Here there are 2 methods recently we are using, AP3 and AP4 method. Now AP4 has more advantages. There is no need of a pre-culture. For AP3 method, we need a pre-enrichment of the hepatopancreas where this bacteria is present. Hepatopancreas should be pre-enriched, should be cultured in pre-enrichment media, and that will help the parahemolyticus to grow more in numbers, so that the number will be sufficient to check the we can get more DNA from this parahemolyticus. We can uncheck the presence of this particular pathogen. And AP4 method, just we can extract the DNA directly from the infected animal, and we can check the presence of parahemolyticus, which is having the PUR-A or PUR-B gene. These primers are developed based on the plasmid PUR-A and PUR-B genes, so that only this parahemolyticus containing this plasmid genes can be detected through these methods. So this is all about the bacterial diagnosis. And we have a set of testing for the soil, water, as well as the pathogens in the PLs as well as in the adult shrimps. CIFRI has the facility to check up. So farmers are interested, you can send your samples, and if you want avail training facility, you can contact us for the same. Thank you.
Thank you, Doctor.
Any question from participants?
Yes, Doctor. Actually, the shrimp is a bottom dweller, Doctor. They don't swim up. Like if the fish is having any disease, at least when it dies, it floats. But the same thing we cannot see with the shrimp. How do we identify that it is dead, and how can we bring it out and see what disease it has? Because we don't know if it is dead or not.
No, usually we used to provide the check trace. It is not, no, we cannot say that shrimps are not swimming in the water. They swim.
They swim, yes, sometimes they come up. That is my next question.
Yes.
But if they die, how do we come to know?
So usually we provide this feed trace we provide.
Yes.
on different corners of the shrimp ponds. So when we give them feed, we used to give in the feed trays also. So
Yes.
we closely observe the number of animals coming into the check tray. What is its color? What are this movement? Is it normal, abnormal? Any changes? This is flocks of we have to observe continuously throughout our culture period. So the observation totally on the check trays will give you an idea about their populations.
Okay.
And
But doctor, it's not that the infected shrimp will come into the check tray. Sometimes it might not.
yeah, sometimes it will not, but you will see on the corners, you can see pink colored. Most of the infections for WSS fever, actually you can see all the sides. Shrimp will come with the stressed, they will come appear like stressed and they will die on the sides also. And
Oh,
if
oh,
the
oh.
number of animals are reducing in your check size, that will clearly indicate you that there is some sort of something is happening in your pond.
In that case, what do we do, doctor? Because I'm not having a pond. I do it in a tank, like an above ground tank. So in that case, what do? We
Now,
discharge all the water.
yeah, exchanges is better options if you suspect anything. But you are telling there are some mineral deficiencies, you have to be prepared. You cannot give a sudden shock to the animals. So you have to pick up the water which is equivalent to the requirements of the animal and give to them. Understand? So sudden shocks you should not give. You should make the water compatible for the rearing conditions at present. Then you have to give it 10%. If it is a flow through, it is doesn't matter. You can do that. But if it is a complete exchange, you have to do a slow exchange only. It is possible.
Okay, no. So on my next question, sorry, if the shrimp is swimming on the top or to the sides, is anything wrong with that? Because what I heard.
Yes. There are certain behavioral changes will come to the animals when they are infected. They will jump. Normally, if when you do a farming, you should closely monitor and observe your animals. They have a normal behavior. Something you are observing which is deviating from that normal behavior will definitely an indication of some sort of infections or stress posing to the animals. There will be erratic swimming. I told erratic, corkscrew swimming will be there for the infections. They'll jump out. So many. No, if we are disturbed, the shrimps will jump out of the tanks. But normally, without any disturbance, stress, it comes out means it's definitely there will be some sort of stress and infections.
Okay.
You can give a sample if you want to test something, you can give a test sample for any of the aquatic labs. They will test you for all the known pathogens usually present.
That's the problem, doctor. In Telangana, we don't know where to send these
You can take
samples.
the samples and give all type of except the bacteria. You can for bacteria you can put it in water and send with the live animals. The live animal transport it is little bit costly if you have to send by air or something like that.
Okay. Okay. Thank you, doctor.
Any more question?
Sir, excuse me. Sir, it is to be said that we have parameter test kits to check the other minerals also,
Yes,
as than salinity.
yes. We have
So, ma'am,
calcium and
can,
magnesium.
hello. Can you please give such any optimum range what is to be all minerals and water?
I don't get your question. It is not audible clear. You are
Yeah.
asking about the mineral constitution in the water.
No, ma'am, no. Am I audible, ma'am?
Yeah, it's audible, but it is little like delayed.
Hello. Ma'am, actually, I wanted to ask
Can you
that
pick down and
we know
send me in the
that
chat
we
so that I
need.
can see?
Hello.
Yeah, please type.
Can you write on the chat box?
Okay, sir.
Can you write on the chat box? Your voice is not clear.
This is a good question. It's a better question.
We are waiting for the question to come
Yes, sir.
in the chat box.
Yeah. Any other questions?
Yeah. What is optimum range of all parameters?
Sir, I have given.
Yeah.
Yeah. I have given in the first itself, sir.
Yeah. That madam covered in the first page. Yeah. These parts madam already covered in the first page.
Yeah.
You can see in the YouTube again. Mr. Ronkit, you can see the presentation in the, if you want to go back, this is available in the YouTube. You can nicely see it.
Yes.
Okay? So I thank Professor to Dr. for her covering two aspects. One is the bacterial disease, as is the water quality. I was finding that how to cover the water quality in the streams because that is very, very important.
Yes.
And that's the prime trigger for the water, the disease in the streams. Thank you, madam. Now we move to next presentation. We are a bit late also, but we don't want to miss that presentation. That is about the feed and feeding management by Dr. A.M.A. Hassan, Principal Scientist, CIFRI, Barakpur. He has lots of experience in the designing of the fish feed for fishes. He has some patents and some products also that is widely sold in India. So without delay, I hand over the mic to Dr. Hassan. Dr. Hassan, sir.
Yes.
The present now.
Thank you, Dr. Manna.
Share enter screen. Share.
Window.
No. Enter screen.
Share. मुझे जहाँ तक लगता है याद है विंडो, बट यू आर डूइंग नो बेटर. ये विंडो. Now you click on share.
Share में लिंक नहीं आ रहा. Shift that computer.
नहीं सर, आप यहाँ बैठ दे सकते हैं, उसको भी लगा देगा वहाँ कंप्यूटर में.
इंस्टॉलेशन कर जाएगा. Yes, I'll be talking on feed and feeding management in respect of environment and fish health. So basically, this training program is on disease and health management. But here I'll be touching few aspects of feed and feeding. Of course, there are many kinds of feed like health foods.
Is there a thing?
full screen. Just slide down or okay.
Sound and video.
No, that is open. That is for that one.
Okay.
It's open. So it's audible.
Yeah, I think I am audible. Very good afternoon. I'll be talking on feed and feeding management for fish growth and environmental sustainability. These are two contrasting things, but we have to take care of both the things, the growth as well as the environmental sustainability. Though last 2 or 3 days we have been listening about environment, about pathogens, about diseases and their management. Today this topic is little different from the last 2 or 3 days deliberation. It is little different. I'll be talking on feed and its management. How feed is affecting both the growth and environmental conditions that I'll be discussing. And we know that in any, as far as fish is concerned, any culture system, if you take, particularly if it is a intensive kind of system, there the feed cost goes around more than 50%. And especially last few decades, there has been a tremendous development as far as fish nutrition and feed development is concerned. And now we have species-specific formulations. In earlier days, it was not there. Common feed they used to apply for all sorts of fishes. Now, as far as freshwater farming system is concerned, the new word has emerged today, that is the cage farming. We have been doing pond farming, we have been doing the brackish-water farming, we have been doing now the new term, that is the cage culture. So that is a very important area because when we talk about cage culture, here the fishes are confined. They are prevented from collecting the natural food from the ecosystem. So the development of feed is to be meticulously designed, precisely made. Then only we can expect optimal growth from the cage culture system. As I just talked about, that there are different kind of culture system. Earlier days we used to consider only the closed water, like the pond, the tank, the raceways. But now today we are talking about the open water agriculture, open water aquaculture, be it in ocean, be it in inland sector. So how we are doing this open water aqua farming? In that respect, either we have to resort to cage farming or pen farming. And in a cage farming, generally we follow the intensive farming system with high stocking density, totally dependent on feed, and along with feed, the health management. And pen is, even today, it is generally we are going for both intensive and extensive. And other system, you all are probably in the business, you know what is pond farming is both extensive, intensive. Tank, of course, it is intensive. And raceways. And some people in the beginning, in the inaugural day, people are inquiring about the RAS system and the biofloc system. So that is also intensive kind of farming. Now, in intensive farming, we have to take care of few issues. Like it demands high stocking density. Then when it is a high stocking density, then there will be high oxygen demand, and there you need a quality feed. Quality feed means it should include all the essential ingredients and including the micronutrients, and there should be perfect balance of energy and protein, then only that feed qualifies as a quality feed. And also a few parameters while discussing, it becomes very subjective. So few parameters we can cross-check, that is the balanced nutrient energy, and it should have good digestibility. That is a big issue in fish feeding system. In the discourse I discuss all these things about the digestibility, and the feed should be good stable, because we are talking, we will be focusing on open water cage culture system. And of course, those who are in pond system, they have certain advantages. Even if the feed is not stable, then that does not matter much, because even if it is unstable, it will be disintegrated and it will remain in the system. And in due course, fish may consume and may not consume. And even if it is not consumed, then it supplements the nutrient content of the system. So that way it is not that much of loss. But in open water system, once the feed is disintegrated, then it will be out of the system, and that feed will not be available for the organism, the fish or any other organism. And since it is an open water system, we cannot regulate the control. We cannot control the parameters. Like in pond culture system, we have been listening about the so many health products, how we can maintain the water quality, even the aeration, the removal of mud, application of so many prophylactic things. So that is possible in a closed water system, but in open water system, that is not a possibility. So having said all these things, the importance of good stability becomes a prime concern for open water farming. And lastly, the waste management. In intensive farming, there is a very important issue, and this last 2-3 days we have been talking about all these things, how to manage waste, so that the water condition remains good, suitable for the fish farming, and it ensures the good health of the organism. Now, I'll not talk about this, just to indicate that how our country has progressed from as far as fish production is concerned, from both marine and inland sector. And in inland sector, we are gradually, day by day, we are improving our yield picture. You can see the globally. Earlier days, it was a capture system. Now, almost it is becoming equal, the culture and capture. So, as the day progresses, our aquaculture system is progressing. So, that aquaculture means feed, fertilizer, medicine, seed, all these things. So when we resort to aqua farming, then we should take into account all these things: feed, health, fertilizer, and good seed, quality seed. And as you progress in aqua farming, there is a growing trend of feeding management, both in developed countries and in developing countries. And this trend has become very prominent in our Indian scenario, because now we are doing both low value staple food and also the high value cash crop. Like the previous lecture was talking about the prawn farming and shrimp farming, and that is a high value cash crop. But in freshwater, mostly we go for low value staple food, like the carp, tilapia, and catfishes. Of course, catfishes means the new catfish introduced in our water, that is the Pangishodon. Of course, and if it is a Etoprustis fossilis or Clarius batrachus, or even if it is a marl, this is all high value fish. And we have been putting feed for all the farming for all these carps, tilapias, and catfishes, and as well as for the high value cash crop like shrimps, and of course worldwide another few species like salmon, trout, yellowtail, seabass. These are high value species, and they are all feed based, and they are cultured under intensive farming systems. And in farm management, we should have the management goal. The management goal, maybe it's a short-lived or it may be long-term. Long-term means it is a sustainable. Our issues, how can we have a successful aquaculture in long-term? And then there are two approaches. One is industrial approach, it is mainly economics. It is focusing on the economics, how the profitability will be more. An environmental concern here, the profitability may not be that much, but we give more importance to towards the sustainability. And before we enter into the feed and feeding subject, we have to understand that feeding behavior and regulation of food intake, because unless we understand the feeding behavior, because there are several kinds of feed available. Some are mash feed, some are sinking pellets, some are floating pellets, some are granular form, and the fish may be carnivore and the fish may be herbivore. And some are bottom feeder, some are column feeder, some are surface feeder. So unless we do not understand the feeding behavior, we cannot precisely provide them the right kind of food. Suppose a bottom feeder, if you give them a floating feed, then it is a futile exercise, and it is vice versa. So we should know before resorting to feeding, we should know what is the feeding behavior and what are the factors that regulate the food intake. Once we know this thing, because why are we talking about this? Because we have to precisely feed the fishes. I will come in the discourse. Suppose what are the existing feeding method? Feeding method either satiation feeding and demand feeding. Demand feeding, of course, is not a very common practice in our country. So we resort to satiation feeding. And there are several disadvantages for this satiation feeding, because fish is an aquatic animal and we do not know how much feed they have consumed, how much they have not consumed, if it is a sinking pellet. So that's why the floating pellet has some advantages over the sinking pellet. Here we can understand the consumption rate and other factors like the nutritional factors. If the feed is energy rich, then there will be less consumption. And if it is energy poor, then there will be more consumption. So more consumption means there will be more fecal matter. And that will deteriorate your water quality. So it should be precisely balanced between the energy and the nutrient content. Then it becomes a perfect feed, and that will ensure the water quality. Otherwise, what will happen? More fecal matter. If it is energy poor, more carbohydrate, then it will deteriorate the water quality. And so there are certain environmental factors. The number one is the temperature, because fish is a poikilotherm animal, and its consumption depends upon the ambient water temperature. As you have observed that during winter seasons there is a reduced feed consumption. And other than that, we have another environmental parameters we have listened in the earlier lecture about the ammonia or nitrites. That blocks feeding. So if that level, unless we control that system, ammonia in the aquatic system, then there will be reduction in feed consumption, and ultimately we end up in poor growth and poor yield. And third is dissolved oxygen. That is a very vital factor as far as fish is concerned. So it should be maintained at optimum level so that the fish consumes its required quantity of feed. And if it is below optimum, then fish will stop feeding. And third is behavioral. If the fish is under stress, then there will be less consumption, there will be less digestion. And then diurnal variation. That is very important. There are certain fishes which feed nocturnally, and there are some fish they feed during daytime. So unless we understand that when the fish or the organism consumes feed, then there will be a wastage of feed, because the feed will be in the system and it will leach its nutrient that will deteriorate the water quality. And the photoperiod. The photoperiod is another vital issue as far as feeding behavior is concerned. And then digestive physiology. That is a very vital as far as feed formulation is concerned and feeding management is concerned. Unless we do not understand the digestive physiology, we cannot formulate a right kind of food and its digestibility. I'll show you a few photographs. the digestive system of the fishes. I'll come later on. Let us discuss about the environmental approach. As we have understood that feeds are the basic input in the intensive farming system, and what happens when the food gets inside the digestive system. After digestion, some part is assimilated, and that meets the physiological need, and then the second part, it's generally the organism they excrete out, and fish they do through gills, through urine, they remove their excretory products, and and in the excretory products there are two components. One is a solid, another is a liquid part, and both these components contains this phosphorus and nitrogen. And probably we have understood about the importance of the role of nitrogen. The solid waste, that is the fecal matter, that gets settled at the bottom sediment, which is generally anaerobic in condition. And if it is anaerobic, then what happens that you have learned in your previous lectures. And this problem is a very localized problem wherever particularly in cages, the cages is a floating structure. And below that cage, or if it is a pond and if it is a broadcast feeding, then it does not remain localized. And throughout the pond ecosystem, this problem will be there. But if it is inside and enclosed in a cages, then the uneaten feed and the fishes will drop just below the cage. So it is a very much localized problem. And these sediments will lead to flux of ammonia and phosphates in the overlying water. And when these fecal matter and unconsumed food they gets deposited at the bottom, then the bottom biota will be changed. So that gives some indication. If bottom biota is changed, that means there is a deterioration of the sediment quality. That's why the benthic population, if there is a change in benthic population. And the kind of feed we are providing to fishes, we know that some are moist feed, some are pelleted feed, and again two types, sinking pellet and the floating pellets. And the feeding method, generally these are the 4 or 5 systems of feeding as they are. Some are broadcasting, some are back feeding, some are tray feeding, some are frame feeding, and automatic or demand feeder. Now, these are the basic things we should keep in mind. And when the feed is formulated, we take help of these raw materials. Some are agricultural byproducts, some are industrial byproducts, some are fish industry bycatch, and some are animal industry byproducts. So these are the generally commonly used ingredients for feed formulation. Now, the question is, these are the ingredients we use in feed formulation. Now, let us consider the digestive system. I was talking about that. Unless we understand the digestive system of fishes, then our feed formulation will not be a perfect, wise one. Let us understand these figures. The top 2 figures are having a defined stomach. That means they have stomach digestion in the process, the digestion process. And last 2 photographs, if you see, they do not have any stomach. So these fishes, they do not have a stomach part of digestion. Now, if we feed these fishes, so 2 categories in general, 2 categories. One is having a stomach, another is they do not have a stomach. But what are the ingredients we are using in feed? If you see, these are all mostly land-based products. If it is a brand, if it is a wild cakes, if it is bone meal or any other products, mostly it is all terrestrial products. And these terrestrial products we are feeding to the aquatic organism. So this terrestrial products is meant for terrestrial organism, and aquatic products are meant for aquatic system. But what we are feeding to fishes mostly land grown, except the fish meal part. Most of them are land grown. So these fish, they are not equipped with the system, be it enzyme system, be it their morphological, anatomical features. If you see, they are not suitable for consuming this kind of food material. So what we should do? We should ensure that even if it is a land-based system, we should focus on its better digestible. We should provide the feed in such a form that it becomes mostly digestible. And if it is digestible, then the assimilation will take place. There will be less of excretion, less of fecal matter. So how digestion and the system, how it influences the environmental quality. So ultimately, if we understand the FCR, and this also, FCR is a just index to understand how it is affecting the fish growth and how it is economically beneficial and how it is also environmentally beneficial. The poor FCR means the poor growth and poor environmental quality. Good FCR means good growth and good environmental quality. And how come we can ensure the good digestibility, good FCR, if we understand the digestive system. So we have to ensure that the food which we are providing to this organism, the fishes, it should be good digestible. How can we ensure? We have to process. This now comes the processing of feed ingredients. And of course, I have not given any formula. I'll just explain here. There are few steps in feed formulation. If you can ensure these few points, like the ingredients, whatever you are using, that should be finely powdered, so that every bit of feed gets in touch with the digestive enzymes, so that it becomes easily digestible. So that's why we have to make it very finely powdered ingredient. Second thing is that it should be cooked. It should be semi-digestive. So some of the digestion part should take place outside the fish gut. So what we can do? We can process the ingredient. First step is grinding, making fine powder. Second step is cooking. Cooking is the first step of digestion. That is the ex situ digestion inside the utensils. Now, once that is done, so fluid is now almost become digestible. So if it now enters into the fish gut, so this feed is now in a nutrients are in a simplified form. Now the fishes can easily digest and assimilate the material, the nutrients from the food, and that will ensure their good growth, and there will be less of fecal matter, and that will improve the water quality. And these are some of the happenings. What happens if we release such kind of feed which is less digestible, and then what happens? And if it is unbalanced, suppose we are giving more of proteinaceous with the hope that fish will grow faster if we give more protein. That is a wrong conception. The fish requires an optimum amount of nitrogen. So more of nitrogen means nitrogen fish cannot store inside their body. They will definitely excrete it out. And once the nutrient, particularly the nitrogen, excretes out, that will enrich the environment, and that will lead to the eutrophication. Eutrophication means it is enrichment of nutrients. And how it comes? Because of its unbalanced feed. If it is more of nitrogenous feed, that will again deteriorate. If it is less of digestible, then it will deteriorate the water quality. So if this thing happens, then what will happen? It will exceed the carrying capacity of the water body. Every system has its own carrying capacity. The dynamics, suppose the nutrient load and nutrient metabolism. If nutrient loading is more, then metabolism will be less, so there will be excess of these nutrients. That will lead to the eutrophication. And eutrophication means more of bacterial load, more of other unfavorable planktons, and that will lead to the poor environment. And you have to spend a lot of money for correcting these bad environments. I just have given one example here. If suppose to produce 1 ton of fish, suppose 1800 kilogram of feed is required, and that means FCR is 1.8. And if that fish contains 47% of protein and with 1% phosphorus, then what happens? So we are providing 18 kilogram of phosphorus and 135 kilograms of nitrogen were feed into the fishes. And of these, 5 kilogram of phosphorus and 30 kilogram of nitrogen will be retained by the fish for their growth purposes, and rest they will excrete out to the environment. Now FCR is less, suppose 1.8 in case of, if it is not 1.8, if it is less than 1.8 or 1.6 or 1.2, what happens? And we have seen that higher the FCR, means more the FCR, then there is a so the protein retention rate is high, a conversion rate, protein conversion rate is high. And if it is less, FCR is less, then it is low. So nitrogen load and phosphorus load will be less in case of better FCR, and if it is FCR is high, then it is more. So these are the things which I wanted to discuss with you, that we have to take care of the FCR. FCR gives the indication of both fish growth and ensures the environmental health. And it is a nutrient loading in case of fish farming through feed. And we have observed that the nitrogen in the sediment it is around 23%, and carbon it is around 29%, and phosphorus it is around 54% in the sediment. So these are the things which you should take care about before feeding. That feed should be properly processed, and it should have good digestibility. And other than that, of course, we are incorporating several additives in the system. I think many of the lectures have covered all these things. When we feed, we incorporate antibiotics or any parasiticides we apply. What happens to the system? That we have learned and listened in last 2-3 days. I will not discuss these things. And what is the remedial or what precaution we should take? So first thing is we should feed the fishes the requisite quantity. So what will happen? Then we can prevent the wastage of feed. And wastage of feed in both, it is both economically and environmentally. It is a deleterious, it is a detrimental. If we give them precise amount of feed, then that will ensure complete consumption of the feed, the given feed. And if even a kilogram of fish is remain unconsumed, so that is the economical loss, and that feed will again deteriorate the water quality. And then we should split the feed we should split the total ration into different portions. So we should have frequent feeding. If we have observed that if frequency is more, it ensures better growth. But one-time feeding, of course, it is though it is a practice, but we have observed that there will be delay in consumption, and if it is delayed, then there will be nutrient leaching, and the growth is poor. But if we split the total ration and give it different times, then there is a better consumption, less nutrient leaching, and better growth. And also we should follow this circadian rhythm that we have observed, that I have discussed in the first second slide, that we should understand the feeding behavior of the fish. So the circadian rhythm is the biological rhythm, biological clock of feed consumption, and not only feed consumption, any other biological system, they follow a circadian rhythm. So the feed consumes the feed at a particular time, and we should understand that time, and feed should be given at that particular time, so that there is an instant consumption, and that will ensure the better digestion. So if we follow the circadian rhythm, then there is a lot of benefits will be there. And if we can follow these 2-3 protocol, then there will be less of phosphorus, less of nitrogen in the system. So I already have discussed about the FCR, how it improves the growth, how it improves the pollution, water quality in the system. And when we select raw materials for formulating our feed, that is a very important aspect of feed formulation. That both ensures the cost effectiveness of the feed and also the nutrient content of it. That will ensure ensure its digestibility and required amount of nutrient from that particular ingredient. So selection of ingredients is very another important factor, and those ingredients should have good digestibility. Unless it may be nutrient rich, but if it is undigestible for a particular species, it may be digestible for a particular species, but it may be indigestible for certain species. And so which fish we are targeting and what feed we are giving, so that matters. Suppose a high protein content, or suppose for example the leather meal or the feather meal, that if we feed include in that for a carp or any omnivorous fish, then they will not be able to digest, because they do not have the stomach part which requires stomach digestion. So we should take care of all these, the selection of ingredients for a particular fish species. Otherwise there will be a lot of nutrients, a lot of pollutions in the system, and that will not give you good growth, rather that will deteriorate the water quality. And lastly, the palatability. The food, whatever the food you are giving, that should have good palatability. Otherwise the feed may not consume your feed, and if it is not consumed, again that will deteriorate your water quality, and that will deteriorate your economic return. And this I have discussed all this. When to feed, we have standardized two things for our freshwater fishes. We have seen that if the temperature is below 24 degrees centigrade, particularly the carps and catfishes like Pangaseum, they do not consume feed. So we should regularly monitor the environmental quality, like particularly the temperature, and we should take care of what temperature and the environmental temperature. Based on that, we should calculate our ration level. And second is the frequency and circadian rhythm I was talking about. We have observed that 8 o'clock and 3 o'clock is the circadian rhythm. And if our feeding time matches with this rhythm, then there will be better consumption of feed, and better consumption means better digestion and better growth. And that will also, better digestion means less of pollutants, less of nutrients in the ambient ecosystem. And with this few areas which I feel appropriate for this lecture series, disease and health management, I thought these are the important areas with the fish farmers we should take care of. With this, I conclude. Thank you very much.
Thank you, sir. Thank you.
Any question? No question?
Hello.
Hi, yeah. Yeah, tell.
Hello. হ্যালো স্যার শোনা যাচ্ছে?
হ্যাঁ শোনা যাচ্ছে।
স্যার আমি তেলাপিয়ার কালচার করি, আমি এতে নিউ টাউনে থাকি, রাজারহাট নিউ টাউন। তো আমি বলছিলাম যে অ্যাকচুয়ালি তেলাপিয়ার জন্য কত পার্সেন্টেজ প্রোটিনের ফিড বেটার এবং কত পার্সেন্ট করে দেওয়া উচিত?
Can you repeat your question?
কত পার্সেন্ট প্রোটিন ফিড দেওয়া ভালো আর কত পার্সেন্টেজ দেওয়া ভালো, মানে 1 কেজি মাছের জন্য কত পার্সেন্ট ফিড দেওয়া যায় থ্রুআউট কালচার?
তেলাপিয়া
আপনি মিউট করে দিন আপনার।
tilapia tilapia is a omnivorous fish. So here the protein requirement in the growth stage it will be less. So maybe maybe you are culturing in pond condition.
হ্যাঁ স্যার, পুকুরে চাষ করি।
Pond condition. We collect lot of food material from the environment. Okay, so in the growth stage, 25% protein is enough for tilapia culture. But early stage, if you are starting from the juvenile stage, then maybe it is 32% protein is enough for tilapia.
আর বলছি ধরুন একটা মাছের জন্য ডেলি কতটা ফিড দেওয়া যেতে পারে? ডেলি ফিডিং
আর যখন আর্লি স্টেজ থাকে তখন সাধারণত আমরা 7 to 8% of their body mass biomass. You have a calculation for your fish biomass in your system. So depending upon fish biomass, early stage it is 7 to 8%, but as we reach to growth stage, then it can reduce to 3%. And if it is a well fertilized, well manured pond, then you can further reduce. Because there will be a lot of periphytic material and they consume detritus. That is the advantage with tilapia farming. So they will feed more on detrital load, organic matter, and the periphyton available in the system. And certain amount of energy and protein they will take from the given feed. So here you can reduce your ration level as well as the protein level if it is exclusively tilapia farming.
Thank you, sir. Thank you.
Hello, sir. For IMC, hello. Sir,
Yes,
I'm Manoj Dey from Korba, Chhattisgarh. Sir, actually, I'm culture IMC. For IMC, I want to know that is there any temperature-dependent feeding system or chart which depends on the temperature of the water, so that we can give the feed according to the temperature of the water.
Yes. As I have mentioned, I see two things in the environmental condition, and also I have discussed about the size and age of the fish. Generally, at the earlier stage, juvenile stage, the protein requirement is higher. In case of major carbs, 32% is good enough for early stage. So and it is also temperature dependent, environmental condition. We have observed calves below 24 degrees centigrade. They cease feeding. They stop feeding or they reduce consumption below 24. But if it is 24 and above degree centigrade, then you can give the full dose of feed.
Okay. Thank you, sir.
You only have to take the environmental temperature. So that is very important thing.
Not the water temperature, right, sir?
Water temperature.
Okay.
Cage culture there is question है क्या? Cage culture which region? If it is starting from Nagpur to Jharkhand, this is a good temperature region, and southern India also. Here the temperature never goes below 24 degrees centigrade. So round the year these fishes can be cultured. But in the North Indian scenario where the temperature falls below even 20 degrees centigrade, in such areas the pangasius culture is very risky. So generally before November the fish should be harvested. And that time there will be less consumption of feed, and simply they will occupy your water body, but there will be no growth. And third thing is there is a chance of mortality, particularly North Indian condition where temperature falls below 20 degrees centigrade. As far as pangasius cage farming is concerned.
Okay, thank you.
Yeah, cage culture feeding again, we have observed that 8 o'clock, and if you go for thrice feeding, that is the best option for cage culture. At number one time is 8 o'clock, then 12 o'clock, then 3 o'clock. These three feeding schedule you have observed, that gives the best growth.
For IMC also, sir?
Yes, IMC we can reduce to, in place of 3, you can go for 2.
Okay, sir.
Any other question? Mr. Srinivas Yadlapalli, you have asked for feeding chart. If you share your mail ID, then we can share with you. I have not included in my presentation. There is a feeding chart for pangasius. Sonit Barma, cage culture.
I'll share you the email address. Thank you, Doctor.
Feeding chart to everybody, sir.
Yes, we can share. Our coordinator, sir, you there are many of your participants are for this feeding chart. If you get the mail ID, I'll share you the feeding chart.
Okay. Maybe either today or Monday we'll share the feeding chart and name of some drugs.
Sir.
Probiotics.
Yeah. Thank you, Doctor.
Okay, welcome.
Sir, whether we will get some manuals for these trainings, that is very, very essential for us, sir. Any training manuals or this kind of thing, sir?
Yes. But sir, we are very sorry that we didn't prepare any manual.
Okay.
Because if you see the presentations, though these are presentations from, most of the experts have very long experience. Although
Right, sir.
For a particular topic, but it's very, very, very diverse.
Right, sir.
From their experiences, the presentations are mostly from their experiences.
Right, sir. Because, sir, it is very unfortunate that we are government employees. The problem is that now we are on the election duty. So whenever we get the time, sir, I have missed out this valuable feed presentation. So that's why, sir, something inputs. If we get, sir, really highly obliged.
It will be there in the YouTube.
Okay. Full.
Full. All 3 days.
All 3 days, the full. Okay. Thank you, sir.
Sir,
Yes.
Sir, actually, I wanted to ask, sir, the species such as singing or all those are Maharashtra, where my location is, it doesn't have any fluctuation of temperature. Like in winter also it don't go below 20 degree.
Below 20 degree, generally after November. After 15 November, the temperature will fall. So you have to manipulate the system. You have to organize your farming management is such that you should exploit the better temperature regime. Starting from mid-February to mid-November. So just 8 months, you get the full range. So your management could ensure your fishes, fingerlings are there in your cage by mid-February. And if you continue till mid-November, your fishes will be 7 kilogram size.
And sir, the bigger the size, they won't have any impact of the intervals, right, sir?
Pardon?
I mean, if the size of the fish is already bigger in, so they don't have any loss in winter, right?
If your fishes have reached that marketable size, then
Actually, the problem is in our state, in our market, only it demands in winter only. When the temperature is down, at that time only the demand is of fish is there, and hence I am doing Singamur. All high catfishes I am doing so. So in market also at one time only 100 kg is most they will accept it, or else they will not accept it. The rate will go around.
okay. So you mean to say that during winter months there is a good demand for catfishes.
Right.
Now what you do, there are two issues. Number one is the chances of mortality. Number one. Number two is the feed consumption. I have said in this low temperature regime, fishes will not consume as much feed as they used to consume when the temperature was above 20 degrees. Okay, you can give them a maintenance ration. Your feed consumption will be less. So what you do, you give low protein diet at a maintenance ration. Okay, 1 to 2 is sufficient if the temperature is low. Okay.
Okay, sir. Okay. Okay, sir. Thank you.
If there is no more question, I'm closing this presentation session. I thank Dr. Hassan for his excellent presentation on the feed and feeding management for environmental sustainability. And now we'll be proceeding towards the valedictory session of this program. And we have sent some feedback forms in your email. You can send the feedbacks. And in that, kindly mention your email IDs. In that email ID, you will be sending the participation certificates. Probably we shall try to cover it by Monday or maximum by Tuesday, because a number of participants are there. So I am directly proceeding to the valedictory session.
Okay.
Hello, sir.
Yeah. Yeah, please tell.
Sir, নাম্বারটা পাওয়া যাবে? আপনার নাম্বারটা পাবে?
হ্যাঁ, আমার ফোন নাম্বার,
মোবাইল নাম্বারটা।
হ্যাঁ 943334
স্যার বলুন। 94334
75913।
75913।
হ্যাঁ 913 913।
হ্যাঁ, ঠিক আছে স্যার, ধন্যবাদ স্যার, থ্যাংক ইউ।
Sir, please share the medicine list, sir. Medicine list and probiotic and feeding charts.
Yeah. That I post in the email. Okay.
Thank you, sir.
Setting chapter
মেয়েরা বললো যে কি একটা কাজ করছে আমি যাবো না, আমাকে তাই বলছে।
একটু দেখে Okay. We are already late. Sir. Of course, sir. We have come to our valedictory program of the 3 days training. We have come to the end of this program, of this 3 days training program of the fisheries and health management in inland waters. It was a tough time, or rather I would say the COVID taught us a lots of lessons. One of them is that how to be online, how to go online. So we tried in this situation how to go online to all the participants, to the farmers, students, other trainings, as well as some of the participants were from the officials also. And we have tried to cover as many chapters or different aspects as much possible. I will tell. And in this valedictory session, I have also requested all our resource persons for their gracious presence. And initially the program was made for 2 days, but directors had told why not to take the help of all other experts from different ICAR institutes and universities. And with his guidance, a tentative topics we could list and send to all the experts, and all are readily agreeable. I thank to all of them. And whatever, ultimately you see that all these topics have come that cover nearly all the aspects, from the viral disease, bacterial disease, fungal disease, parasitic disease, streams, and then finfishes, nutritional management, then from the private entrepreneurs, and including the booming topic biofog, etc. So I hope this will be very much useful to all the participants. In this training, we had most of the resource persons from the outside, and from Dr. S. Odhikari sir from ICR CIFRI, Dr. P.K. Sahu from ICR CIFRI, despite his illness, he was present, he presented today. And Dr. P.K. Pradhan from NBFGR, Dr. Professor T.J. Abraham sir from West Bengal University of Animal and Fishery Sciences, Professor Godadar Das from West Bengal University of Animal and Fishery Sciences, Mr. D. Borman from Aquaductor Solutions, Dr. A. Sahunje from ICR SIVA, Dr. S. K. Atta from ICR SIVA, Dr. Vidya Rajendran from ICR SIVA, Dr. Panigrahi Dr. from ICR SIVA, and today, now in this at this point, I request our acting director, Dr. S. Samanto, to take the lead in this valedictory session. With us today, we have Dr. Godadar Das with us in this podium. So I also request Professor Godadar Das from West Bengal University of Animal and Fishery Sciences to take chair as the guest in this program. Thank you, sir. We could not, what director sir suggested at the initial beginning, maybe some practicals, but in this program we could not attend organized practicals. Maybe in the later on, maybe some online practicals that we have to find it out how to present it. And of course, all the 3 days presentations are there full there in the YouTube. Already the link has been shared with you. Now every day, at any point of time, something around 30 to 40 participants were there. But on an average, you have tabulated around 80 to 90 participants participate in the Google Meet, and in the YouTube, there is around 350 to 370 views daily, including today. Now, I request Dr. Samanto, Acting Director, to kindly say something.
Myself or other field, till then.
Okay. And now, I request our Extension and Training Cell in charge, Dr. A.K. Das, to say few kind words. Sir, it catches.
Today, our general acting, Dr. Samanto, the course director, Dr. Mana, one of the best friend of CIFRI, I said CIFRI was going to University of Animal and Fishery Science, Dr. Godadar Das, and our heads of divisions, Dr. B.K. Behra, and Dr. M.A. Hassan, and Arun Purohit and Dr. Sumit Kumar, and our fellow colleagues of CIFRI, and the participants in this 3 days long online training program on fish health and management under All India Network project on fish diseases. So, in most occasions, I was there in observing all the presentations, and in most cases, most of the presenters are from outside. ICR institutes, ICR CIFRI, ICR SIVA, and ICR NBFGR, and they're quite potent and very resource persons who delivered and tried to answer all the questions came from our participants. And very pertaining to that, the biofloc systems, Dr. Panigrahi from ICR SIVA nicely presented, and not only deliverations but also because because it's a very burning issue in this present day, and our honorable Fisheries Minister, Sri Singh also under Pradhan Mantri Matsya Sampada Yojana, that putting most attention towards the backlog systems to be in place. So it will take time, but very robust technology. Hope in coming days it should be working with this joint intervention of these knowledgeable resource persons, field workers, and the professor fishers, and followed by other fishers and other fishery extension officers across India, district fishery officers, assistant director of fisheries, and all the line departments and ICS systems. So the 3 days deliberations, theoretical classes, because we are online, and no option for practicals, even though the photographs, field-based photographs, more than 85% photographs are from field-based, and which were nicely presented to accommodate our training participants, so that they would be viewed those presentations, photographs, slides in a more vivid way as if they are in the field. So in coming days also, CIFRI would go with such training programs, so long as pandemic not yet completed, that phase is going on. So with all this, I extend a very warm gratitude to the team the running this show, and inviting all the very renowned resource persons across India, mostly from ICR institutes as well as universities pertaining to fisheries and thank you all.
Thank you, sir. May I request Dr. B.K. Behra, Principal Scientist and Head of Division, to say a few words.
Thank you, Dr. Manna. I hope that 2 days training program might have gone well, and 3 days training program might have gone well. And as per our schedule, as Dr. Das told, many things has been told regarding the burning issues like backlog in RAS and other disease management aspects. I think it will be great use to all of you. And we will have also future collaborations, and in future, it's not like that this training program is over, so everything is over, so we'll have collaborations with CIFRI and in future also. And I want to compliment all the organizers for organizing such a wonderful training program despite this COVID situation and despite their different problems, but they have organized. I will compliment each and one of them. Again, thank you.
Now may I request Dr. M.A. Hassan, Professor and Head, to say a few words, sir.
Again, good afternoon. The Director in charge today, Dr. Samantha, our respected guest, Dr. Gadhav Das, and our heads of the divisions and other colleagues of CIFRI, and dear participants. I understand that 3 days is not sufficient, but Dr. Manna has made it a capsule form so that all aspects of health management and disease management can reach to you. And 3 days, if you carefully listen this program, and these programs are also available. He is repeatedly mentioning that even if you have missed some lecture, these are all available in the YouTube. So later on, you can leisurely go through those slides and those words spoken. I think that will help you in managing your fish and your fish farm. Of course, this is a very good effort, and we are relentlessly working for the fishery development of fishery sector through our knowledge-based guidelines. And today, even during this COVID period, when all the sectors are showing negative growth, but fish sector has maintained a sustained growth. That indicates our farmers are managing their farms, and we are just supplementing their knowledge. We are enriching their knowledge. So future farming will be knowledge-based farming. And this kind of initiative, I hope your future participation and better management of your farm and better yield. With these few words, I congratulate all of you, those who have taken part in this training program. Thank you and best wishes.
Now Now may I request Dr. Nag to say a few words. Sir,
Thank you, Dr. Manna. First of all, I compliment the course coordinator, Dr. Manna, and his entire team for nicely organizing and successfully completing this training program. And also congratulate all the participants who have attended this 3 days training program. And I hope that really you have learned something, although you knew something already, but the training added some knowledge. But all the trainings, all these things would be successful if you stay or keep in touch with the subject. Because we go to attend trainings many times, and after that we don't touch it or don't culture it, and then we forget it. But you have to be constantly in touch of those who are in the field and the students who are studying it, so that you don't forget and you further try to enhance your knowledge. So then the training will be very successful. And CIFRI will be there. All the resource persons are there. Contact is there. So keep in touch with them for any further problem or any further queries. Then there will be two-way process of communication. The resource persons will also respond to your queries. Then the learning will be very useful for you in future. So I congratulate once again and best wishes for your future career. Thank you.
Thanks, sir. Now I request Dr. Arun Pandit to say a few words.
Thank you, sir. Respected Dr. Samantha, today's lecturer, Dr. Jidas from Kolkata, guest today, all the HODs, scientists, and all the participants. So hope in these 3 days you have learned many things regarding fisheries management, and hope also that many of your questions may not be answered because of lack of time. So you can have touch with them. All the experts are there, so they have phone numbers, so you can get your questions answered anytime. And I think thanks to the online platform, you can see the experts, you can hear the experts from Chennai, Bhubaneswar, Kolkata, Barakpur, at a time, and also you can anytime, anywhere, you can again watch, again learn from the YouTube platform. So it's a good use of online platform, and I wish you all the best, and hope that whatever you have learned from this training program, you will apply them and you will get benefit out of them. Thank you very much.
Now may I request our research persons from other institutes, Dr. S. Odhikari from ICR-CIFA. Sir, are you there in the online? No, sir is not there. Yeah, Dr. P.K. Sahu sir from ICR-CIFA. Dr. P.K. Pradhan from ICR-NBFGR. Or whoever there, Professor T.J. Abraham sir, Mr. Borman, Dr. Abunje, Dr. S.K. Otta sir. I was seeing Dr. S.K.
Yeah, I'm there.
Yeah, sir. Yeah, Dr. Abunje sir, please tell something. Yeah. Yes, sir.
Hello?
Yes, sir, we can hear.
This is Dr. Otta from Siba, Chennai.
Okay, sir.
It was very good interaction with the participants, and I hope I have given something to them. And if anything they have doubt on, they can contact me also. And thanks to organizer for arranging such a beautiful training program online. I'm sure it will be very beneficial to many of the participants. And again, thanks to the organizer and all the participants. Thank you very much.
Thanks, sir, for your kind time and kind effort, sir. And Dr. Abunje. You are not audible. Dr. Abunje, you are not audible.
Is it audible now?
Yeah.
So there are some different settings. Generally, we use Microsoft Team. So anyway, so it was very nice program arranged. Even I did not anticipate such a good participation by farmers, maybe some students or state administration and all. So it was nice talking to you all. And thanks to Manna sir and other CIFRI administrator, SICs, HODs, in charges for arranging this program. In the presentation, anyway, my email ID is shared. If anybody is having any doubts regarding the topic I had discussed, they can communicate to us. And today's program, I think somebody had asked regarding SIPA marks. So I have shared our SIPA director ID and also my ID. So they can, I hope they will be contacting us. So thank you, everyone. It was a very nice program. Maybe in future we will meet again frequently in this kind of online platform. Thank you all.
Thanks, Dr. Avinde, for your kind time and effort. Dr. Panigrahi sir, are you there?
He is not there. He is busy with another talk.
Okay.
Participants.
Now, I request the inputs from participants. One or two participants kindly say something.
Yeah.
Inputs.
Yeah, I'm Srinivas from Hyderabad. The online training program was very well designed and organized by AICAR. We thank all the scientists and doctors who shared their valuable researches, their knowledge with us with appropriate information. It was our pleasure meeting all the distinguished scientists and doctors, and we hope these programs will continue further. And I thank Dr. Manna for the coordination done for all these 3 days. Thank you so much, sir.
Anyone more participant? More remarks from participants? Yeah. Sandhya madam?
Good afternoon, sir. This was a lovely presentation, sir. I mean, so many of my doubts have been answered. Actually, I had started biofloc farming last year, March, and the day 12th March, I had done my stocking, and from 22nd March, we went into lockdown. So, unfortunately, I lost my culture. But I have learned a lot from this presentation of these 3 days, and it was really great. I mean, most of my questions have been answered, but I will still disturb all the doctors and professors. Please don't mind, because I want to do many things in aquaculture. So I'd like to thank Dr. Manna and for organizing this. It was lovely and beautiful. Thank you so much, everybody.
Thanks, madam. And now, और कोई कुछ बोलना चाहेंगे?
Respected सर ये ट्रेनिंग प्रोग्राम के दौरान मैंने बहुत कुछ सीखा है। इस लॉकडाउन के दौरान मेरा फिशरी एक्सपर्ट का एक नौकरी था, वो गवा गया। तो फिर मैं फ्रीलांसिंग की तरह हर पॉइंट पे अगर फार्मर मुझे बुलाता है तो फिर मैं उधर पे जाके उधर पे मेरे पास कुछ टेस्ट किट है, तो उसी हिसाब से हम उसको यूज करते। हाँ, तो वाटर पैरामीटर ऐसा कुछ हम डिवाइस चेक करते हैं। तो उसी हिसाब से बहुत सारे डिजीज हमको मतलब फील्ड पे हम डायग्नोस करते, तो उसका फुल रेमेडियल हमको वैसा कुछ नॉलेज नहीं था। तो इसी हिसाब से जब ये ट्रेनिंग प्रोग्राम हम आज अटेंड किया तो बहुत सारे क्वेश्चनर्स जो था, तो मैं ये फिशरमैन को मैं बता सकता जो ऐसे ऐसे हुआ है। इन फर्दर ये जो आप जो वीडियो किया है यूट्यूब में, ये हम थोड़ा प्रैक्टिस करके भी फिशरमैन को ट्रेनिंग कर सकते हैं, जो उसका क्या क्या प्रॉब्लम है और उसके हिसाब से कौन सी दवा हम तालाब पे यूज कर सकते हैं। तो मैम और सभी सर सभी को धन्यवाद ज्ञापन करता हूँ इसी हिसाब से। तो ऐसा ट्रेनिंग प्रोग्राम आपने ऑर्गेनाइज किया और हम लोगों का तरह हम जो फिशरी स्टूडेंट है और जो भी एंटरप्रेन्योर है, तो उसी हिसाब से बहुत अच्छा हुआ। हम बहुत सारे ज्ञान आज मिला है और भी आप लोग का थ्रू और भी कॉल करूंगा और भी ज्ञान चाहिए, हाँ, रिगार्डिंग इस फिश डिजीज एंड हेल्थ मैनेजमेंट। थैंक यू सर।
पवन जी आप कुछ बोलेंगे?
हाँ नमस्ते सर। हेलो।
हाँ यार।
हाँ सर नमस्ते सर, सभी को प्रणाम सर। जो 3 डेज की ट्रेनिंग प्रोग्राम थी सर, बहुत अच्छा था सर, और सर काफी कुछ सीखने के लिए मिला सर। सर थोड़ा सा हिंदी में होता तो और ज्यादा कुछ अच्छे सीख लेते सर, लेकिन बहुत ही अच्छा लगा सर।
हम लोग आगे देखा जाएगा, हम लोग पूरा हिंदी क्योंकि हैदराबाद है कुछ होने से है, हम लोग को हिंदी आती है लेकिन मिक्स हिंदी आगे देखा जाएगा और कैसे।
थैंक यू सर, सभी को प्रणाम सर।
Thank you. Now I request our guest today, Professor Gadadhar Das sir from West Bengal University of Animal and Fishery Sciences, to say a few words.
Very good afternoon to all in this galaxy of all the heads of the departments and director of CIFRI, the Premier Institute, and just one to three things I want to mention here. Is last 3 days perhaps Dr. Manna has troubled you like anything, but I can say that it is not a troubling time or a trouble matter, but it's you have utilized or fruitfully utilized the time. You have not wasted the time. So I am seeing the chart, and you are from 9:45 to 5 o'clock. So entire day almost with a little lunch break, you have attended the training programs. So last 3 days, so you are with these resource persons. Of course, resource persons are changing, but participants are same. So definitely it's a troublesome matter, but you have accepted, you have presently heard all those things, and definitely it will help you, it will reward you, it will give in the feedback definitely to you. And another thing is that this is totally a practical based thing, and Dr. Manna has tried a lot to arrange it to deliver something to you people. So he has compacted the program in such a way that at least it has covered almost all these different topics, and covering the resource person from the different places, starting from institutes, universities, and all the experts. They are masters in their own field, and they have tried their level best to deliver the things to you people. And some feedbacks what I have got that regarding the language, that is Hindi, and some practical things, some practical aspects. Definitely I will request the coordinator to arrange more programs and invite you people also once again to have practical based or field based knowledge from CIFRI, and equip yourself for future consequences of disease occurrence in the field. And one more thing I'll tell you, to arrange this program, perhaps Dr. Manna has spent not less than 15 days to 30 days to arrange all these resource persons, to arrange all these topics and get it done in a smooth way. So I should thank him, and I should thank all his research scholars who has helped him a lot, and also the staffs of CIFRI who has helped him to make this program a very successful one. And last but not least, I should thank the director because who is the key person in all these activities in CIFRI. Of course, today he is not there. He has given the charge to Dr. Samanto, who is also instrumental in many other activities of CIFRI. And he is here also today with us, and he will speak also something to you. And with these few words, once again, I am thanking you, all these participants. And I will tell you one other thing. These participants, this training program is a liaison only. So you just keep liaison with all the resource persons among yourselves also, so that you will enrich still more. Science is a never-ending process. You will learn so many things, and gradually, and also in future days. With these few words, once again I am thanking you, all the organizers, and particularly the coordinator and his team, and the director of CIFRI, to arrange such a beautiful program in such a synchronized way, inviting the resource persons from different corners and inviting the participants from different corners and making in one platform. And that is possible due to COVID, because that gives us an immense opportunity to be in this technical age or technical era. With this, let me thanks once again to all the participants and also organizers, and thanks to all. Thank you.
I sincerely thank Professor Das to grace this occasion and to say kind words in this regard. Now I request our acting director of today, Dr. S. Samanto, and as well as the head of division, to say a few words.
Good afternoon to everyone. Our leader, Dr. Basantakumar Das, Director CIFRI, is out of station. He is on journey now. Otherwise, he could participate in the online mode also. He has really given all the facilities and freedom to organize this beautiful training program. Our today's guest, Dr. Gadadhar Das, professor from West Bengal University and Administrative Sciences, all our head of divisions, Dr. Hassan, Dr. Behra, Dr. Nag, Dr. A.K. Das, Dr. Orun Pandit, the training organizers, Dr. Manna, Dr. A.K. Behra, and all the team. It was really a nice occasion that a large number of participants varied from maybe from northeast to the extreme end of south took part in this training program. COVID has created a lot of problem, but probably this is an opportunity when such a diverse group is taking part in this type of training activities. I took part in the inauguration program also. There were a lot of thinking that how the training program will be. There were a lot of confusion among the participants in their mind that what they will learn. I feel that by spending these 3 days, their time is not wasted. They have learned a lot. As they have given in their feedback, they have learned a lot out of this. I sincerely thank all the persons who took part as the resource persons from different institutes, starting from CIFA, CIVA, West Bengal University Animal Fishery Sciences, and other institutes. I sincerely thank all for giving opportunity to organize this beautiful training program. As the participants have told in the first day, that they would like to learn about the different diseases due to some agents involved, maybe viral, bacterial, fungal, whatever it may be. In addition to that, they had also concern about the different physicochemical parameters, their impact on different diseases, and also in extreme cases, some of the physicochemical conditions that create disease also. I feel these sectors have also been nicely covered by the expert member like Dr. S. Sudhikari. He is very experienced also in that field, in this field. This is not the end of this training program. Maybe this is the beginning of this diverse group. In future, we can go with all these persons in a beautiful way to solve some of the major issues of diseases in the open water field. I thank sincerely to the organizers. Dr. Manna has took a lot of effort and initiative with his team, Dr. Bera and all, to beautifully arranging this program. In addition, there is another one group also led by Dr. Behra, who are doing well also to address the issues on disease management in open water fisheries. Actually, this is basically a challenge. When there are so many problems regarding the antibiotics, their misuses, abuses, and many times in our country, when the farmers mostly listen the dealers regarding selection of the different antibiotics or your medicines for controlling the diseases or adverse conditions, this is really a challenge because many times they get misguided also. So let us join hands together to build a nice era in future. Thank you all.
Excuse me, sir.
Yeah.
Sir, is there Panigra, sir, is going to be there again? Actually, I wanted to ask some query, sir.
I could not follow. और एक बार बोलिए.
I said, our Panigra, sir, is going to join us again, or actually, I wanted to ask something about backlog.
डायरेक्टली कांटेक्ट करिए।
हाँ, आप थोड़ा डायरेक्टली कांटेक्ट करिए उनका नंबर से।
ओके सर, ओके।
उनका नंबर दिया जाएगा, you can directly contact.
Okay.
I have it. Okay, sir.
They also regularly organize the training programs on BioClock.
Right, sir, right.
Okay, please follow the CIFA website. It will be there.
Okay.
Now I request Dr. A.K. Bera to say vote of thanks.
इधर आ जाइए।
Thank you, Dr. Manna. This is my proud privilege to propose vote of thanks on behalf of organizing committee as well as CIFRI. I first thank to our dynamic director, Dr. B.K. Das, and convener of this training program for his continuous encouragement as well as guidance to plan this training program. We must thank Dr. Hassan and Dr. S.K. Nag as co-convener and course director of this program, Dr. S.K. Manna, for meticulously planning this training program and designing the different type of lectures. And we sincerely thank the research person from outside the institute who have given their lectures in their field of expertise. I must take
The names: Dr. S. Adhikari from CIFA, Dr. P.K. Sahu from CIFA, Dr. P.K. Pradhan from NBFGR, Dr. T.J. Abraham, Dr. Gadadhar Das from West Bengal University of Animal and Fishery Sciences, Mr. Devtanu Burman from Aqua Doctor Solutions, Dr. S. Aunje from ICR Siva, Dr. S.K. Ota from ICR Siva, Dr. Vidya Rajendran from ICR Siva, and Dr. A. Panigrahi from ICR Siva for delivering their lectures and sharing their expertise with the participants. And our internal resource person, CIFRI scientists, who have also delivered their lectures. I must thank to all the participants who were with us since last 3 years and interacted with the experts and participated in discussion in depth. I must thank in-charge director Dr. Samantha and all head of the divisions and in-charges to be present in this occasion of valedictory session of this particular training. Today's guest, Dr. Gadadhar Das, who is also present in this occasion. I must thank to in-charge training and extension cell and his team for proper management of the hall. And I thank Mr. Sujit Chaudhary for photography in the inaugural as well as valedictory session. I must thank Mr. S.K. Sahu and Mohammad Naim for arranging the online system to conduct the training program, and Dr. Raju Goitha, who was also coordinator of this training program. He has also delivered his lecture, important lecture in cage management. I must thank Mr. Nilimesh, Sauvik and Sanjeev for suitably creating the online training system and feedback form and creating the e-certificates and continuously monitoring the system and making the training program successful. Thank you. Thank you all.
The training program ends here. I thank you all.
Thank you so much, sir.
Thank you.
Thank you.
Thank you. Thank you. Thank you, Dr.
Thank you so much.
Thank you.
Thanks to Vera also.
Thank you, sir. Thank you.
Thank you, sir. Thank you.
Extension.
Sir, thanks a lot.
না আজকে